Good afternoon, everyone. There may be a few people that are continuing to join. Well, I think some of us are on Zoom every day, if not a new call every hour. Not everybody is in that position, and there may be a few people that are going to join us a little bit late. But I wanted to welcome you to the PEGS Town Hall Forum. Could I have the next slide? So one of the things that we wanted to do today actually is to share the fact that our name has changed. So what was the EPR is now called PEGS. And I like to think about this in some ways as the EPR has grown up. I think that we're really very different than we were at the beginning, and we're going to tell you a little bit about that as we go through today. We want to talk about some of the scientific accomplishments, what's on the horizon for PEGS, as well as answering some of the frequently asked questions, and then opening the questions up to you. So today, we have the participants in our PEGS study who have been invited to this town hall. And in addition, we have invited individuals from the NIEHS. So this is not broad, but we really wanted it to hit the people who would be the most interested in this. Could I have the next slide? So I wanted to start and talk about initially a few introductions. So I'm Jan Hall. I'm the co-PI of PEGS. I'm the clinical director at NIEHS, and what that really means is the director of clinical research. I'm a senior investigator, and I also am chief of the clinical research branch. I'm an endocrinologist, but I'm very interested in how the environment affects our health. A lot of the environmental toxins are, in fact, endocrine disruptors. And we also have to think about some of the other exposures, like our lifestyle exposures, that also affect our health to a great degree. Also with me is Allison Motzinger-Reif. Allison is the co-PI of PEGS. She's an associate investigator and chief of our biostatistics and computational biology branch. Allison, is there anything else that you'd like to say?
No, thank you for the introduction. I'm so excited to be working with this cohort. As Jan said, I'm chief of the biostats and computational biology branch. I'm a statistical geneticist, is probably the best description of me. And I have a long-standing interest in trying to understand how genes and the environment and their interactions play a role in disease risk.
Great, thank you. Next slide. I also want to introduce David Fargo. David is part of the PEGS Executive Leadership Committee, and he's the director of our NIEHS Office of Environmental Science Cyber Infrastructure. And a big part of that also relates to the privacy of your data. And so David has been just incredibly helpful. In addition, we have Charles Schmidt, who is the director of the NIEHS Office of Data Science. And Charles is also a member of the PEGS Executive Leadership Committee. PEGS also has an internal advisory committee, as well as an external advisory committee that is made up of experts in really trying to understand how the environment affects our health. Things have come a long way in that understanding, and it's always wonderful to have additional advisors. We are also wanting to set up a participant advisory committee. So for those of you participants who have received our newsletter recently, you will have noticed that we have put out a call for volunteers who might be interested in our participant advisory committee. and Vida, who is also joining us today, along with Jen Emerson. They're a very important part of our support for the study. Vida is going to put the information up in the chat so that if you are interested in joining our participant advisory committee, we would just love to hear from you. Next slide. So we're going to have a couple questions as we go through today. And the first one on the next slide is, how many total participants have enrolled in PEGS? A, 5,000 to 10,000, B, 10,000 to 15,000, C, 15,000 to 20,000, or D, 20,000 to 25,000. So the poll is being put up now. So give your best guess. Fantastic. I think we can end the poll. And Nathan's going to, well, actually, yeah, Nathan's going to show us the results. And as you, next slide, please. So I feel as though some of you must have read the website or heard us talk or actually read your newsletters. So the total number of participants that have been enrolled in PEGS is C, 15,000 to 20,000 individuals. So we're really, really excited by that. And the majority of our participants are from North Carolina, so we're a really unique cohort because of that. Now, PEGS has actually enrolled 19,672. Could I have the next slide? So just a little bit about how PEGS started and where we are now. So it actually started, almost 2 decades ago. I mean, actually over 2 decades ago. And as I mentioned before, it was formerly called the Environmental Polymorphism Registry, or the EPR, and many of you will have joined the study as the EPR. It's sponsored by the intramural program of NIEHS, the National Institute of Environmental Health Sciences, and it was intended as a long-term project to collect DNA from a diverse group of people in North Carolina. As I mentioned, we have about 20,000 participants who enrolled in PEGS, and we're in regular contact with about 50,000 of those. If you happen to be on our town hall and might be interested in enrolling after you see some of what we're going to present today, please know that enrollment is ongoing, and I'll show you information on how to enroll toward the end of this session. Next slide. So over time, our participants have provided data in a number of different forms. So we started just collecting DNA, but over the years, we've also added a number of surveys to collect phenotypic data. So that's really data about your health, as well as exposure data in our cohort. And we have information from over 9,000 of our participants. The kind of information that we have collected is just general demographic information, family medical history, your own medical history, lifestyle factors, occupational exposures, as well as your address histories. The address histories have been really important because what that allows us to do is to be able to use public databases that have information in them about what's in our air. So the particulate matter in the air, the ozone in our air, also what's in our water, how close we are to, to sites that may have toxic waste in them, how close we are to green space. So that's been really incredibly important to add to the survey data that we have. We've also had the opportunity to do whole genome sequencing. And whole genome sequencing has now been conducted on almost 5,000 of you who agreed to this. One of the things I promised was that I would talk to you a little bit about what you yourselves might be getting out of this. And for those of you who agreed to have whole genome sequencing done and in whom we have that information, we also are looking for variants in our genes, in your genes, that might confer risk of specific health conditions. If you have been sequenced and you have a variant that does confer risk, you probably have already been contacted to provide us additional information, and also so that you can speak with our genetic counselor and be referred if needed. For the next slide, please. So why the name change? Now, over the period of time that the study has been in existence, we have collected, as I mentioned, really a lot of different kind of data types. So we have really multi-dimensional data that relates to our genes, that relates to our environment, and that relates to our health conditions and any diseases that we might have been associated with. So I've introduced, so initially, you know, PEGS was EPR, it was a registry. And a registry just means that information is primarily sitting there. And that's not really what you signed up for. And what we wanted to change our name to a real study was to really recognize that we are in a really good position now to be able to use the data that our participants have provided. We also have a lot of new tools. So I mentioned to you that Dr. Motzinger-Reith is the co-PI of this study. And combined with the computational skills and innovation of investigators like her and her colleagues and trainees, we can do so much more with the registry. We can be so much more than a registry, rather. You know, what we really can do is begin to deliver on the promise of new discoveries that link the environment to human health and that can determine how the environment and our genes interact. The longer term goal of PEGS is to be able to provide the data and methodologies that will allow our physicians and each of us to be able to understand the genetic and environmental factors that contribute to our health and those that are fighting against us, resulting in chronic and often debilitating health conditions. The term precision environmental health has been coined to describe this goal. And again, I think the health piece of that is really important because we're not just about diseases, we're actually about promoting health and helping people prevent diseases. Next slide, please. So on to our second question. So which is the most common self-reported disease or condition among our PEGS participants? Diabetes, high cholesterol, asthma, or seasonal allergies? So Nathan, are you showing the results now? Okay. So again, you're right, it's seasonal allergies. And as I found out when I moved to North Carolina, never having been bothered by these before, this can be a really big deal for so many of us. So again, they're reported in about 40% of all of our participants. Next slide. All right, so now we want to talk a little bit about some of our scientific findings. Previously, we were really able to study things kind of in a small way. Many of the investigators at NIHS are really interested in a gene or set of genes that can help them understand the mechanisms of disease and how those things work. And we have been able to use PEGs and the information in PEGs to be able to help those investigators move their science forward. And of course, the reason behind the science is through understanding the mechanisms of disease, is to really help us determine potential treatments for those diseases. So Dr. Garanciotos has published a study which showed us that distance to roadways increases the risk of asthma in our PEGS participants. And it does so to a greater degree in individuals with a specific genetic variations. Next slide. And Dr. Dmitry Gordinin has shown that while there are breaks in our DNA that occur in the skin, as well as in many of our other organs with normal aging, exposure to ultraviolet radiation dramatically increases this. And this paves the way for potential malignant changes. So again, it's really important for us to begin to understand what the effects are of some of the things that we expose ourselves to and that we are exposed to. And Allison is going to move on to the next couple of slides.
Thank you, Jan. Yeah, I got a few other studies I want to highlight and make sure everybody is familiar with. So we've got a few very recent publications that are work that we've been able to do with this large data and integrating these different data types that Jan has talked about. So we have a great study where we actually showed that association of the distance a participant the distance to a concentrated animal feeding operation, or these swine concentrated industry operations, is associated with immune-mediated diseases, and particularly for individuals with genotypes with certain immune genes. So an interesting putative gene-environment interaction was discovered there. We've also, in another recent study, led by a postdoc, Dr. Eunice Lee, was able to look across all those questionnaires that everybody here filled out to try to find exposures that are associated with cardiovascular diseases and looking for shared exposures across particularly atherogenic cardiovascular diseases. And really were able to reaffirm some known associations and discover some new risk factors as well, including blood type with cardiovascular disease. Next slide, please. We also have a recent publication led by a postdoc, Melissa Lowe, who really did some sophisticated geospatial modeling to understand how mixtures in air pollution, as Jan said, we're not just looking at ozone, not just looking at PM 2.5, but trying to understand how the complex mixtures that we are exposed to are associated with disease, and found some important associations with autoimmune diseases specifically. And then another recent study that I want to highlight builds towards those goals of precision health that Jan mentioned earlier, where we were able to look across all of the exposure data that we collected in those questionnaires and compare it to the genetic data that everybody so generously provided. So we built risk scores. We looked at if we built risk scores looking at your genetics across the genome, how well could we predict your risk of type 2 diabetes, versus if we built built risk scores over the exposure data. And we showed that the poly-exposure, so multiple exposure risk score, was more predictive than the polygenic risk score. And even if you added poly-exposure data onto genetic data, that environmental exposure data always provided a really important lift in how well we could predict that disease outcome. These are some examples of some of the first work we've done. We're expanding these approaches we've built to other diseases and phenotypes of interest, and looking at other questions with this data. Right now, we're really digging into those GIS-based exposures Jan mentioned, where we're linking addresses and address history to sort of the databases of exposure estimates. Next slide. So I get to ask one question too, and you guys are really good at your homework. So we'll see how we do here. But next question, what's the average age for PEGS participants? And this is at the time of taking the survey questionnaires. So I'll launch another poll and we'll see what the group's guess is. Great to see things coming in. We're ready to close it off. We've got good participation here. Next slide. So this group is right on it. C was the right answer. So the average age, we're looking at the health questionnaires, is 49.7. So again, you guys are right there. Next slide. Sorry, one more. Great, Jan, I'll pass it back over.
Great. Thanks so much, Allison. And these are just some of the studies that PEGS has been involved in, and I really want to, I really want to thank the entire PEGS team, in addition to all of our participants, for allowing us to begin to move this science and this environmental health science in all its complexities forward. So what's on the horizon for us? So the first thing that we've been working on, and something that we've recently asked our participants to allow us to access, is in our collaborations with UNC and Duke. So as I mentioned, all of our participants almost are from North Carolina, and UNC and Duke are clearly large healthcare systems across the state. And we've been working with them to be able to capture things like health conditions that may not have been present when you initially completed the questionnaire, and also to provide lab tests and sometimes medication history that will allow us to confirm some of these diagnoses. So none of this will be done without your participation. And I'm going to talk a little bit later about data security, because I know that there's a lot of concern about health information as well as our genetic information. And we'll be talking about that. The other thing that we are now doing and are very much in the middle of is what's called epigenetics. So that while our genes are fixed, there are aspects of our environment that actually can change how our genes function through changes that are referred to as epigenetic changes. And what this is going to allow us to do is to actually look at the exposures, at your exposures, related to these epigenetic changes, to see what exposures appear to have the most dramatic biological effects as measured by epigenetic changes. The other thing that we're doing, and we're going to be reaching out very soon to try and call back as many of our participants as are willing to participate to be able to get additional blood samples. So as I mentioned, we really collected blood for DNA when the study started way back in 2002. But now we can do a whole lot more in terms of our science than we could then. And with additional blood samples, with urine, with things like house dust, we can determine the toxins to which we are exposed and how these toxins might affect our bodies. Could I have the next slide? So I just want to finish this part of our presentation by thanking our participants. It's just what you have done and what you have contributed is just so amazing and really is so helpful. There are many reasons that people choose to participate in studies in general, and specifically those of you that may have chosen to participate in PEGS. It could be because you're generally interested in how the environment affects health, and that's obviously incredibly important. It could be because there's a connection to your family history, or it could be that you just really feel that research is an important part of how we can contribute to knowledge that will not only help us, but will help others. So thank you. A number of you, I also neglected to mention when I was talking about some of the studies that our bench scientists have done. A number of you have already come back to the clinical research unit to participate in these studies. And I just want to thank you guys as well, for again, going above and beyond. And it's incredible. I also wanted to mention that by maintaining contact, by completing surveys, by providing specimens, and by updating your data PEGS itself is able to continue to move our science forward and also to provide you with updates on future events and activities, as well as the highlights of some of the work that we're doing. Next slide. So I want to move now to a few questions that we've heard a number of times. There's only 4. And then what I'd like to do is to open it up for you to ask your questions. Next slide. So the first slide is a really important slide and one that I alluded to a few minutes ago. And that is, how is my data kept private? How is it kept protected? So the first thing is that things like your name, your date of birth, we don't collect social security numbers, so that doesn't count, but other kinds of information is removed from all of the data and all the samples, so you are known by a number. The second thing is that we have unique codes that are assigned to information. A key is created. This key is kept in a password protected file on secure servers under layers and layers of security that Dr. Fargo supervises and makes sure that we are more than 100% compliant with. So the PEGS PIs and staff are really the only personnel with access to this information. And frankly, the only reason we access the information, I never have done it, Allison never has done it, is when we are doing our recontacting. Because at that point, we want to be able to reach out so that you can let us know what's going on with you. Next slide. The other thing is that sometimes we will be asked to share information, your data, not your name, not your age or birth date or any of that, with other investigators. And if you have agreed to that, This data will be stored in, again, a controlled access data repository that's only going to be able to be accessed with approved credentials by people who have IRB approval in general. And IRB is Institutional Review Board that really looks after human research, essentially, and all the regulatory aspects of human research. And so that's an important part of it. And again, everybody who is provided access to this data, to these data, has to sign a confidentiality agreement, again, even though your name and your birth date, et cetera, are not included in that. And finally, we have a certificate of confidentiality that's in place through 2035. So, you know, that's going to be a little bit of time. And what this certificate of confidentiality provides is that researchers cannot be forced to disclose any information that may identify participants, even by a court subpoena, in any federal, state, or local civil, criminal, administrative, legislative, or other proceedings. So we can't do that. So this is a big deterrent. And we would use this certificate to resist any of those kinds of demands, except in cases where you, the participant, has given written permission for this to occur. And then, of course, we wouldn't stop that. Next slide. So the next question is, what's being done with our DNA? So back in the earlier days of understanding human DNA, which was when our study was actually started, we initially were able to look at tiny little pieces of our genes, of all of our genes. So maybe a tiny little piece on a specific gene. But now what we can do is get a lot more information from looking at all of the exons, which are the parts that actually code for the proteins that are critical for our body to function. And we can also look at genomes, which is everything in the entire gene, including all the control regions and all the junk and the stuff like that, that doesn't, where nothing happens. And so this DNA, again, is shared in databases. So this is DNA that's in fact, it's not the DNA itself, it's the code that comes out of analyzing the DNA. It's shared in databases so that approved researchers can read your DNA and then they can make determinations on health and disease. So as I mentioned, those of you who contributed, who agreed to have whole genome sequencing done, we shared the information from that with very close collaborators at the National Genome Institute, who then have the capacity to do all of the quality control and also have all of the genetic counselors to provide information to you. The second thing that sometimes is asked is, what about cell lines? So cell lines are actually created to provide an endless source of your cells and DNA for research without us needing to call you back for additional samples. Now, it's really important that the cell line is not used for cloning. It's not used to grow artificial organs or organisms, and it will not be sold or used for any commercial purposes whatsoever. It really is just a resource so that if we run out of the DNA that you provided through your blood sample, that there will be another source for us to be able to continue the research that you have participated in. Next slide, please. So the next question that we've been asked is, so what's so special about PEGs? right? There's a lot of databases out there that have information on individuals' DNA. And oops, sorry, we've gone to the second bullet here. And there's also a lot of databases that have a fair bit of information on the environment. But we're quite unique because we now have both. And we can look at the effects of the environment, the effects of genes, as well as that combination, the effects of mixtures, as well as your genes on health. The other thing, if we go up to the first bullet, is that PEGS has a very diverse cohort. In fact, there are 62% women in our cohort, and 30% of those are 30% of our participants are Black, which is overrepresented in terms of our state. And there's also a real range of income and educational levels. And so it really is very interesting from that perspective, and again, quite unique in terms of the kinds of cohorts that are out there. And then again, I mentioned about thanking those of you who have come back for future studies. And the other thing that's really unique is for those of you who choose to continue to give us their information. We can also provide you the opportunity to participate in other studies. You are not required to, but that opportunity exists. Next slide. And my final question is, how am I compensated for participating in PEGS? So the first thing is that PEGS provides compensation that is based on the samples or the questionnaires that are completed. This is something that is, again, regulated by the Institutional Review Board at NIH, and the amount is guided by their policies and guidelines. The other ways in which you're compensated, and I hope you feel the love, is that the investigators who have used PEGS, those of us who have really been trying to move PEGS forward, are just so incredibly thankful for your participation and really for giving of yourselves to help us understand how the environment affects our health. And I also hope that you get something from it as well, in terms of knowing that you're participating in something that's really important. So could I have the next slide? So what I want to do now is to move to the questions that are in the Q&A. I think that Nathan has sent a note to everybody asking that you put your questions into the Q&A. And I think Jen and Vita are going to start us off and let us know and read off the questions that kind of as they have come in.
Yeah, so our first question is, what determined who received whole genome sequencing?
I can jump in, Jan, if you'd like, a little bit of a break looking over some of these questions. So there were a few selection criteria, the main one being, of course, only those who consented to it and were willing to share that information. Then we selected individuals that had provided the most health and exposure information. So we tried to make sure we were genotyping those that had answered the health and exposure and exposomics surveys to the maximum. Yeah, those were the two. Like, so you had to have consented, and we wanted to make sure you had the maximum amount of data available.
Thank you. We have received multiple questions about whether there is any possibility of collaborating with other studies, such as All of Us or other genomics cohort studies.
So absolutely, we are already working with some other cohorts and are open to any new relationships, opportunities for collaboration, for replication. Anything there is wonderful. To answer more specifically the questions about the All of Us program, we have really worked to build some very deep connections with All of Us. We did a workshop with NIEHS and the All of Us research program last summer about how to incorporate environmental data into the All of Us cohort, and that was guided by some of the things we've learned, the opportunities that you guys have provided with the samples you've given us in PEGS. So I really feel like it's going to be a deep collaboration that we have between PEGS and All of Us. And I'll give one just sort of tidbit on, of course, All of Us has an amazing sample size and diverse representation across the US, but they actually have less than 1,000 individuals in the state of North Carolina. So when we think about sort of the size of PEGS relatively and what we can find out specifically for our local community, we are not that small. Like I said, we've got many more participants than All of Us to sort of understand the environmental diversity and impact here in North Carolina.
If I could just,
I'm a native North Carolinian. This is really important to me. This is the water I've been drinking. This is the air I've grown up in. This is really special for me to get to work on a cohort at home.
yeah. And just in case there's any concern, we are not going to share participation like anything. We may send something that might show you how you could particularly sign up for All of Us, but we're not going to share names. We're not going to share contact information. We're not going to do any of that. So just to make sure that, there aren't any concerns in that direction. John, I wonder if you could stop sharing so that we don't have to look at our little Pillsbury Doughboys for quite so long. Thank you. Okay.
Right. The next question is, will me being involved in PEGS hinder me from being involved in other research studies?
The answer is absolutely not. And I know that that's something that people sometimes are concerned about. I think that the main times when it's concerned is if you are involved in a study and you're asked to take a medication or something like that. But for PEGS, we generally, unless you're asked to come in and do a study where we'll let you know way ahead of time whether there's a concern, and there's always a possibility of a washout time. You're not going to be prohibited in any way, shape, or form.
Okay, great. Are there plans to collect information on psychosocial stressors such as perceived discrimination?
Yes, in fact, there are. And so we don't have that included in our questionnaires at the moment, although we do have a fair bit of information about your local environment that we know can be related, things like green space, schools, and things like that. But yes, this is a really important question. And I think we've become increasingly aware of the importance of those kinds of social stresses that relate to experiences of racism. So yes, thank you for that question.
We've had a few questions about how people can still participate if they don't live in North Carolina any longer, or if they move out of state, does that affect their participation?
First of all, if you don't live in North Carolina now, it's totally fine. It's just that it's a primarily North Carolina cohort. You may not be our first choice for calling you back because for people past a certain distance, we need to pay transportation, and we don't have that deep pockets, but we would love you to continue. And there are a lot of studies that can be done now, and I think COVID helped this to some degree, where we're actually sending out packages and having people send those back. And you can collect your house dust that way. You could collect urine samples that way, and you can even go to a local lab and have blood collected that way. So please stay engaged.
Next question. Do you know of any studies with the DNA data in alpha-gal syndrome?
I don't personally, honestly, certainly not within anything we've done in PEGs.
Yeah. One of the things, though, for the person who asked that question, there's a rare disease network at NIH, and that might be something worth Googling and being contacted. The Rare Disease Network, it's really centered at the Genome Institute at NIH, but there are a lot of other institutes, in fact, who are involved in that, including us for some diseases, but we are not, that's not one of the ones that we're doing for this disease. And it's not one of the ones that is, I mentioned about reporting back results, and there are a specific number of genes that we specifically look at that the American Association for Genetics has deemed to be the important list. And these are ones that are very actionable and relatively common. But I would suggest that you look at the NIH Rare Disease Network and see if you can find out more information there.
Okay, great. We've received a few questions about people wanting to update their contact information. We replied to some of you, but just please reach out to our study team at the email address that Vita put into the chat, and we will get that updated in the system for you. The next question is,
by the way, please do that, because we really want to be able to continue to be in contact with you.
Mm-hmm. The next question is, if I don't hear back about my incidental findings, do I assume I don't have any?
If you have had whole genome sequencing done, the answer is yes, but you don't know whether you have had whole genome sequencing done. If you would like to find out that information, you can also send a note. I think Vita, it's the same. Vita, Jen, it's the same contact information. And what we will do is determine whether your sample is one of the ones that was sequenced, and we'll let you know. If it was sequenced and you have not heard, that means that there aren't any findings that are the kind of thing that you would want to know, so that you could get more frequent mammograms, or so you could be screened for colon cancer or those kinds of things.
Okay. Will the slides be available somewhere for attendees? I know we were going to post a recording, but someone is asking about slides.
I don't see why not.
Okay.
So I think that we can put those on our website along with our recording.
Okay. Next question is, what will the participant advisory board do?
Help us a lot. And the reason that I'm saying that is that we want to know things like, do you want newsletters? Do you want more town halls? Do you want, how often do you want us to contact you? And really to help us provide information on that, as well as other things that participants feel that we could do. Clearly we can't do everything, and it has to be within scope in a way, but a lot of it is that we often have these questions, and we don't want to drive our participants nuts, but we want to keep them informed, and we want to make sure that we don't lose track. During COVID, we lost track of a whole lot of people because the kind of migration across the United States was huge. And so, we don't want to do that. We want to make sure that we can stay in touch if you want us to stay in touch.
I'll also add on to that. We would really appreciate participants' guidance in some of the scientific questions
Yeah.
We ask, right? You guys know what you're exposed to, what is worrying you, and what is impacting your health. So on top of just sort of engagement at the, we're hoping not to drive you too crazy level, we also know that you guys know what questions we should be asking and trying to answer scientifically. So hope to have that level of engagement as well.
Yeah, no, that's really important because, you know, NIH is a big place. NIH has an extramural part, which is giving out the grants, and an intramural part that we're part of. And the scientists are amazing. And we don't have to just collaborate with ourselves. We're actually hoping to be able to collaborate. I mean, we already do, but we also want to be able to collaborate even more with other scientists. And so some of the questions that you might have are ones that potentially other scientists would be really interested in, possibly using our exposure data. So thank you, Allison.
I think this is our final question. You mentioned the diverse cohort that PEGS is, and the question is, was there any targeted effort to make it that way?
There actually wasn't. And it's interesting. There were in the early days of PEGS, between about particularly like 2003 to 2005 or 6, there were a lot of kind of recruitment fairs across the state. And that's really where the majority came from. Now, the majority of our participants are from that triangle area, from, you know, Durham, Raleigh, Chapel Hill, but we also have really significant numbers of participants from the eastern part of the state and from the western part of the state. So in general, we know that women tend to volunteer more than men. So for any of the men out there, step to the plate, if you like. And we're just really thrilled that we have been able to engage the African-American community. And, you know, I think that a lot of the issues with the environment are so incredibly relevant across the board, and for a number of minority groups in the state. So whether they're Native Americans or African American or Hispanic. And so I think that, again, I'm you know, we're going to begin more serious recruiting again, and I think those are some of the things that we're trying to do. So this is very much a convenience cohort, as we call it. All of us is one that has specifically tried to target specific groups, but that isn't something that we have done. So if I guess, are there any more burning questions, Jen?
One more question just came in that says, does anyone ever age out of the study?
Never.
That's a quick to answer. Yes.
Never.
So that, I think that's all of the questions we've had.
We want you forever. All right. Could I have the next slide, please? So I wanted to just thank you for attending. Thank you to my colleagues on the call for all of their input. And I'm just going to leave a couple of the URLs for two of our websites up for a couple of minutes as people begin to sign off. The first is the general Join a Study one, and PEGS is prominently featured on that. And it links to our general website, but we also have a separate website that you should feel 100% comfortable going to, because it's got lots and lots of good information on it. So I want to thank you again. And with that, we'll wrap up.